The laser point. Hey guys, welcome back to Pre-AP Biology, DNA History, Structure, and Replication. This is part 2 of the first part of this lecture. So today we're going to be talking about the replication of DNA. And the intro picture here is basically showing you this is a pretty complicated process. We have a lot of different players here, all sorts of different enzymes. We have things going in different directions and whatnot. This process is happening in your cells right now at this very instant, and it's happening at a very, very rapid pace. Okay, so let's get into this whole process that's going on. First thing, remember, DNA replication occurs during the S phase of the cell cycle, and S stands for synthesis. That is when the DNA is going to be replicated. So we've learned about how Jim Watson and Francis Crick described the structure of DNA, and as I have said many times in the past, structure determines function, and the structure of DNA leads to its function, which is the molecule of heredity, and also as the million-dollar blueprint, if you will, for the instructions for building all of the proteins that make up your body. So it is stored in the nucleus in eukaryotes, where it is protected from other types of degrading enzymes, and it's a double helix structure, which also keeps it nice and stable compared to the single-stranded RNA molecules, which are your cheap dime store copies that can be readily disposed of. Okay. So one of the important pieces of evidence that Francis Crick and Jim Watson had when building their models was the base pairing ratios of Irwin-Chargaff. Remember, Irwin-Chargaff did not say that A paired with T or C paired with G. He just suggested that the ratios were similar. If you had 30% of A, that meant you had 30% of your DNA was composed of T, and 20% would have been composed of G, and 20% would have been composed of C. That's all he said. It was Jim Watson, who came up with how the bases paired together. Okay, so again, structure determines function. If you take a DNA and you have one strand of it, and you split the DNA down the middle, so you now have two strands of the DNA. Each strand acts as a template for building a new strand. So looking at this particular picture here, you can see we're starting off with the DNA, which is dark blue. The first step in replication is to separate those strands. And then we're going to add new nucleotides, light blue in this case, in order to build a new or two copies of DNA molecules. And if you notice, each copy of DNA molecule is composed of one new and one old strand. This is called semi-conservative replication. Fully conservative replication would mean that we would start with a strand of DNA all dark blue, and then we would end up with one strand copy that was all dark blue and one copy that was all light blue, and that just wouldn't work. So semi-conservative replication is the model that researchers used to figure out that was their hypothesis, that the replication would be semi-conservative, and they did prove it through a series of experiments. All right, so replication in DNA was worked out in prokaryotes, i.e. bacteria, mainly because they are simpler. They have much less DNA. They have only one chromosome inside of their cells. It's a single circular chromosome, and it's just easier model to work with versus having something like human DNA, which has 46 linear chromosomes. Okay, so again, it was worked out in prokaryotes, and it's a lot easier. As you can see, we start off on the left-hand side of this diagram. We have a single point of origin. That's where replication will start. And then the replication forks, the DNA is going to be split, and replication will continue in both directions until we get to the termination of the replication site, and we end up with 2 copies of DNA. It's a little bit more complicated in eukaryotes for a number of reasons. 1, there are multiple chromosomes. 2, each chromosome is much larger than a single prokaryotic chromosome. So in order for DNA replication to occur in a timely manner, we're going to have multiple points of origin. Each chromosome is going to have multiple points of origin, and it leads to a couple of issues, which we'll see in the next slide. So here is a picture of both a scanning electron micrograph and a cartoon of DNA replication in eukaryotes. And as you can see in diagram number 1 at the top, there are 3 points of origin, or origin of replication spots there. That leads to 3 different replication bubbles, and we're going to have a total of 6 replication forks. Okay, and there's going to be some issues going on, which we'll get to a little bit later. But the main point is that this allows for DNA replication to occur in a timely manner in much more complex eukaryotic organisms. Okay. DNA has direction, and where that direction comes from the sugar molecule of the nucleotide. Remember, a nucleotide is the building block or the monomer of DNA or RNA, and it consists of 3 parts. We have a phosphate, a 5-carbon sugar, and a nitrogenous base. Well, the nitrogenous base is attached to that C1' carbon, and you can see that on the right hand side of the diagram. And let's go in a clockwise manner. We have our 2' carbon, then our 3' carbon, and the 3' carbon is going to have an OH group, or an alcohol, coming off of that group. Then we go to our 4' carbon, and then our 5' carbon. And if you notice, the 5' carbon has a phosphate molecule attached to it. So the phosphate molecule is attached to the 5' carbon. The OH group, or alcohol, is attached to the 3' carbon. And the nitrogenous base, the A, T, C, or G, is attached to the 1' carbon. That little hash mark means prime. Okay? And this is going to give DNA a direction due to how nucleotides are added. Do you remember how what the process is called that that builds macromolecules? We'll see. Okay, so here is where the problems start to come into effect. If you notice, look at this diagram. Let's start on the the left-hand side. We have the new strand of DNA. And you can see we have the 5' end. What does that mean? That means the very top molecule on that is going to be a phosphate because it's attached to the 5' carbon. And if you look at the bottom part of that, it says there you can see the OH group coming off of that sugar, and that's called the 3' end. So the 3' end always has an OH group. The 5' end always has a phosphate. How do we add new nucleotides to this molecule? You got it through the process of dehydration synthesis. That means we have to take a water molecule out. Well, where's that water molecule coming from? Well, if you notice, the 3' end has 2/3 of the water molecule right there, the OH, and there is a hydrogen on that nucleoside triphosphate. This is also a building reaction, an anabolic reaction, an endergonic reaction, which means it requires energy. Where is that energy coming from? It's coming from that nucleoside triphosphate. That's very similar to ATP. Besides adenosine triphosphate, which you are very well familiar with, there's guanine triphosphate, adenine triphosphate, that's ATP, thymine triphosphate, and cytosine triphosphate. each one of these nucleoside triphosphates has its own energy incorporated to it. And we can only add them on to the 3' end. So DNA can only grow in what we say the 5' to 3' direction. Okay, and this is all going to be catalyzed by an enzyme called DNA polymerase. And if you also notice when you look at this picture is that the template strand is running in the opposite direction of the new strand that's being built. The new strand goes from 5' to 3' reading from the top, whereas the template strand goes from 3' to 5'. Alright, so that's what Francis Crick discovered, is that these DNA strands are exact copies of each other except that they run in opposite directions. It's like if you took two pencils and held them up next to each other, and then you took one, you had eraser to eraser and point to point, but then you take one pencil and turn it around 180 degrees, so you have the eraser on one end of the left pencil, and at the right pencil the eraser is at the opposite end. Okay, so that's kind of similar to the way DNA strands run. And again, this is an important fact that is going to cause some problems when we start to replicate the DNA. Alright, so a couple of different things. DNA has another peculiarity, and that is that the enzyme RNA polymerase cannot attach itself to naked DNA. Okay, not sure exactly what the evolutionary baggage is there, but it can't. And so what has to happen before DNA replication happens is that an enzyme called primase is going to come in and lay down a short 3 to 5 nucleotide sequence or starting point for DNA polymerase to attach to. And once that's on there, DNA polymerase can come down, attach to that RNA primer, and it can start to build the new copy of the DNA based on base pairing rules. And so you can see this. We have primase comes in, it lays down the red primer in the 5' to 3' direction, and then DNA polymerase is going to come in, attach to that RNA primer, and start adding new nucleotides once again in the 5' to 3' direction attached to that primer. So we need to talk about the different enzymes in DNA replication, and you need to write these down in the order in which they come in. There's 5 enzymes. The first enzyme is helicase. What does helicase do? It attaches at the replication origin point, and again, in eukaryotes there are multiple replication points of origin, and the helicase is going to unwind the DNA about 1 to 200 base pairs. It unwinds the DNA. Primase. We just talked about primase. Primase will now come in, and it's going to lay down a short RNA primer, once again in the 5' to 3' direction. DNA polymerase 3. DNA polymerase 3, look at the word, polymerase. Okay, it's an enzyme, ASE, all of these are enzymes. What's a polymer? It's a it's a whole bunch of monomers stuck together to make a large macromolecule. DNA polymerase takes nucleotides and turns them into a DNA polymer. Okay, so polymerase 3 comes in, and it's going to build the new DNA strand once again according to base pair rules. Our next player is going to be DNA polymerase 1. What does it do? It comes in and it's going to replace the RNA primer with DNA. So it's going to take out that RNA primer, add a DNA molecule, or replace the RNA nucleotides with DNA nucleotides. And the last enzyme is ligase. And what ligase is going to come in and do, it's going to stitch together all of the different fragments of DNA. They're not quite joined. The sugar phosphate backbones are not quite joined up. And ligase is the enzyme that's going to come in and kind of stitch them all together to make one complete DNA molecule. Alrighty, so let's look at our DNA molecule up here. Okay, DNA polymerase is the molecule responsible for elongating the DNA. What molecule is responsible for unwinding the DNA? Answer that. Okay, and then what's the next molecule? What's the next enzyme that has to come in and lay down something? Answer that. Alright, so once we have that helicase has unwound the DNA and primase has laid down the primer, then polymerase is going to come in, and remember, we can only make the DNA in the 5' to 3' direction. So in this case, helicase is moving towards the left-hand side of your screen, and that's going to cause a problem. And if you look at this, we have a leading and a lagging strand. What's the difference? Okay, the leading strand, the new DNA molecule, is being made in a continuous process. Why? Because it's being made in the 5' to 3' direction. But in the lagging strand, that's not happening, because as the helicase moves further down, or I should say further to the left, okay, the DNA, if you notice up there, that leading strand, the 5' to 3', the lagging strand, the 5' to 3' direction, is going away from the replication fork. And so the lagging strand has to be made backwards. And when it does that, it's going to create a whole bunch of small fragments of DNA that are called Okazaki fragments. And again, we'll watch some videos of this, and hopefully that'll make it a little bit more clear. So here we go. Here's all of the players together. There's a replication bubble, because remember, DNA replication is occurring in both directions at the point of origin, and we're just looking at the left-hand bubble and that smaller left-hand fork from that smaller upper picture. Okay, and then we blow that up and we look at the overall direction of replication. We see helicase, the green blob, moving towards the left. We see the RNA primer and primase coming in, laying down those red primers. We see DNA polymerase 3 on the top strand. That is, which strand? Is that the leading or lagging strand? And then we see DNA polymerase on the bottom strand. It's moving, it's a little bit further away from the RNA primer, and it's coming up upon another RNA primer. Alright, and then we see a little bit further to the right on that bottom strand, DNA polymerase 1 coming in and replacing the RNA primer with DNA. And then we see another enzyme, DNA ligase, coming in and stitching together those Okazaki fragments. So if I was you, I would go copy or click on some of these websites below to look at this process in real time. We will also look at some of these in class. Make sure to have some questions ready for when we come to class. Okay, a lot of times problems can happen during replication. We can make mistakes, okay, and those mistakes need to be repaired. Normally mistakes aren't made too often, okay, because DNA polymerase 3 is kind of a proofreader. It makes sure that the correct nucleotide is there. But that being said, mistakes still occur. Not very many. About every time the cell divides, about 30 mistakes are made. Now that may seem like a lot, but there are 3 billion base pairs. So 30 mistakes and 3 billion base pairs is a minuscule amount. Even computers can't copy their own code that well. So this is a very, very efficient system. And polymerase 3 can come in, and there's a couple of DNA enzymes. One's called a nuclease enzyme. It will recognize a mistake, it will cut it out, and then DNA polymerase will refill that gap, and then DNA ligase will come in and seal it. And that's how repairs are made. Some people, you may know some people who have skin cancer. You know, I myself have spent a lot of time out in the sun, yet I've never had skin cancer, whereas other people can get it very quickly. Oftentimes that's because they have a mutation in the nuclease enzyme that cuts out the damaged DNA, and that's what helps to prevent skin cancer. And if you have a mutation in that particular enzyme, you're not going to cut out the damaged DNA, and that's going to lead on to more problems, i.e. cancer. Well, that's it, guys. Hopefully you enjoyed it. That's 20 minutes.